So I'm a postdoc at UCSF, and I'm going to talk to you a little bit about the work that I do there, as well as how I got there. So I grew up in the border town of Ciudad Juarez, Chihuahua, and that is next to El Paso, Texas. So living in this border town, it's a very unique experience because it's a mixture of both cultures. So you have the Mexican culture and you have the American culture, and they kind of both intersect in these two cities. So it doesn't really feel like you are either in the US or that you are in Mexico. It's just a blend. So it's a very unique experience, and I loved it. So I was in high school. I studied all the way up to high school in Mexico. And so while I was there, there was a teacher. She was great, and she was the one that kind of started me in the path of science. So she was a chemistry professor, and that's when I realized that I had a passion for chemistry and I really liked chemistry. And so I knew I wanted to do something with chemistry, but I just didn't know what it was. So then I did my undergrad at UT El Paso. So I went across the border and I did my bachelor's there. So here, like I said, so I did all the way, study all the way up to high school, so in Spanish. So this was my first barrier. I kind of had to, take going from classes in Spanish to going to classes in English. So this was its own challenge. I had to kind of, while I was taking classes, I had to translate what the instructor was saying to Spanish and then translate it into English so that I could write. So it took a while, but fortunately I was able to overcome that, and I'm here now. But being at UTEP, there's about 80% of the total population of students are Hispanic. So it really pretty much felt like home. It was still part, of what I was studying. It was a mixture of cultures, and it really felt like I was at home. It really didn't felt like I was studying at, in a university in the U.S. And so this is going to come relevant in a minute, and I'll tell you why. But while I was at UTEP, I was a major in chemistry. And so one of my professors suggested that I should apply for the MARC program. I don't know if you guys have heard of a MARC program. Any hands? No? Okay, so the MARC program, or the RISE program for that matter, these are programs that are aimed at increasing diversity in the sciences. And so they pretty much fund you for 2 years for you to do research. And then I think in both cases, they include doing a summer internship at an outside university. So this was the first time that I had heard of this, and so I decided to give it a try. And it turned out that I was accepted. So this was kind of the stepping stone into my into my science career. Here I was doing research, and I was getting paid to doing research, so it was amazing. They kind of showed me how to apply for a grad school. So I was going, and I knew at this point I wanted to do chemistry, and I knew that I needed to go to grad school. So from here, I graduated with a bachelor's in chemistry, and then it came down to where was I going to for grad school. So I graduated in December, which meant that grad school usually starts in August. Which meant that I had this whole semester. So I decided to do a postdoc here at LBNL. And so this is where my husband was doing his PhD as well. So I was there for 6 months, and so I started applying for different graduate programs. I think I applied to about 5 or 6 of them. One of them was Berkeley. I obviously wanted to go to Berkeley, right? I was already here, my husband was here, so it made perfect sense. But when I applied to a very, very small program. So small, they only accepted 1 person. That person was not me. Then we kind of went back to the drawing board and what are we going to do? And so I had an interview at UT Southwestern, and I had a really great experience. And while my husband was also an undergrad at UTEP, he had done a summer internship at UT Southwestern. So he knew the associate dean there. So we started talking to her, he transferred, and we both are at UT Southwestern now. So I went from being in the majority, 80% of total students were Hispanic, to being the minority. So this was a cultural shock to me. At this point, I really, I didn't realize that it was going to be such an impact on me. I missed home. I did not understand half of the time what the classes were about. And so it was, at UTEP, I was prepared to do chemistry, but I think that my other fellow classmates were taking advanced courses in organic chemistry, so I felt a little bit behind. And so what I was really frustrated, my first exam, I pretty much failed. It was horrible. I was like, what am I going to do? I'm already here. And so I just started talking with my other peers, and I realized that I was not the only one. We were all feeling the same. We were all missed home, and we just, it was difficult. Even if they had taken more advanced organic courses, it was difficult for them too. So that really helped. That opened the communication, and then we started having study groups. We started asking our professors to help us understand concepts. And so it was through this that I was, at the end, thankfully, I was at the top of my class. So then here, I obtained a PhD in analytical chemistry in the lab of Dr. John McMillan, doing natural products research. And so here, one of the advices that I would like to do, if any of you are interested in pursuing a career in science and a PhD, is that I would try to find a lab that you feel not so much that the project is important or that you like the project that you will be working on, but that you actually have chemistry with your PI, with your professor, the professor that you will be working with. Because at the end, this person is going to be in your professional life for a very long time. So you want to have a good connection or chemistry with this person. And so that's what happened with him. He has supported me now and continues to support me. He's a great mentor, and I'm really grateful to him. And so now it came the next question. So I want to do a postdoc. Where do we go? No, we have a two-people problem. But my husband was graduating first. And so I was like, well, you kind of move away with me to go to Dallas, so it's your time to choose where we're going to go. So we did, and he ended up coming here at Stanford. And so I knew while I was at UT Southwestern, I went to a SACNAS conference. This is a Society for the Advancement of Chicanos and Native American in the Sciences. This is a very, very big conference. So you have universities from everywhere, and so I was here. I ran into a poster that of the URACTA program, and so the URACTA, this is a postdoc fellowship, and what this the aim of this fellowship is for students to gain access or training in teaching. So while you are doing research, you are also teaching. And so I applied to two of these programs. One is at Stanford and one is at UCSF. And so there are different flavors of these fellowships throughout the country, and there are different requirements for each of them, but this is the overall idea of these URACTA programs. And so I applied and I got in. So then it was a matter of choosing my lab. I completely changed fields. So I was an analytical chemist, and I switched to a lab of it was chemical biology, but right now I'm mainly doing molecular and cell biology. And so I knew, that it was going to be a challenge, but I really wanted to do. I kind of wanted to compile what I knew from my graduate work and kind of combine it with molecular and cell biology techniques that I could apply later. And so while I'm going through this whole process, I realized that I share a passion for mentoring and teaching, and that I want to expand the diversity efforts in the science. And so Now, my trajectory is going more on the administrative path more so than the professorship. This is where I'm at now, and so I'll talk to you a little bit about what I do, okay, in the lab. And you're welcome to ask any questions. You interrupt me, and I'll be happy to answer them. Yes?
Hi there, thanks for coming. My name's Juarez, I'm Vice President of Construction. So I did have a question, I'm glad you opened it up. What did you just say, that you're on the administrative path? Is that what you
That's what I want to
That's
do.
what you want to do.
That's what I want to do.
I think that a whole bunch of us don't know what administrative path means.
Yeah, so what I want to do is more on the kind of diversity, outreach, educational research type of path. So it's limited in the sense that there are not a lot of jobs that, it's a very specific profession. But that's kind of the path that I'm looking for.
Running those programs, project managing,
Exactly.
exploring funding,
Yeah,
making those
that-
programs more viable.
Yeah, that focus on diversifying the STEM fields.
Thank you so much.
Yeah. Okay, so I'm going to start with some basic concepts so that we are all on the same page, and you can further understand what I'm going to go into more detail when I talk about my research. And so first, I'm going to talk to you about, this is a cell and the different components that are part of the cell. So here we have the nucleus, and this is where your genetic information is stored. This is where you have DNA replication and transcription. Then you have this that it's right outside of the nucleus, and that's the cytosol. This is where what translation happens and protein degradation occurs as well. Then you have, and I'm sorry, you can barely look at the pointer, but this, whatever it's on the outside of the nucleus, and that you have these kind of black dots, that's the ribosome. And the ribosome is what translates mRNA into protein. And then you have the ER, that's what it's kind of surrounding those black dots. And this is where it's a very important process, and this is a process that I'll be focusing on throughout the talk. And this is where you have a process called co-translation translocation. And what happens is that a protein is being made while it's being translocated inside the ER. And I'll go into more detail, okay? And then here you have the Golgi apparatus. The Golgi apparatus is kind of the ferry of the cell, and that's what sends the different proteins into the different destinations, okay? And yeah, so for now, I think that's all you need to know. And then here, this is just the central dogma. So you go from your DNA template, and that DNA template gets transcribed into an mRNA, and then that mRNA gets translated into a protein. Now the protein, it's pretty much what carries out the actions of your DNA. So what are proteins? Why do we care about proteins? This is going to be kind of the main focus of the talk. And so you maybe have heard of proteins in your milk. Well, there are proteins in your milk, but they are kind of the worker bees of our cells. And so they are found in our hair, in our nails. They are also involved in the transport intracellularly. One example is the one that I just told you about, the Sec61, or the translocation. That you have these pores that allow proteins to go in and out of the different compartments of the cell. Then you also have antibodies. Antibodies are proteins, and these are the, when you're sick, then your cells produce antibodies to protect you from pathogens. And these antibodies are proteins. Then proteins are also enzymes. Enzymes are important for cell function. They regulate a lot of critical chemical transformations that otherwise wouldn't happen without them. So as you can see, they are very important, and they are the molecules that put the cell's genetic information into action. So this is the endoplasmic reticulum, or rough ER, and this is kind of the block dots that we saw around the nucleus. So there are two types of ER. We have the rough ER, and the rough ER is just represented by the block dots here. Those are the ribosomes. And then you have the smooth ER. And the difference is in the rough ER, you have proteins that are being made that are going into the membrane of the cell, or that are going outside of the cell, okay? And then in the smooth ER, you have other proteins that are, not proteins, but lipids or hormones that are being made, okay? And these block dots, like I said, they're ribosomes. So now, if we look at the actual process of making a protein in the ER, it looks something like this, okay? So you have this little, what is called a signal sequence. Well, first you have your mRNA, right? And that mRNA, it's being translated by the ribosome here. And then you have this, what is called a signal sequence, and it's just a short amino acid sequence, about 10 or 30 amino acids long. And so this signal sequence tells the cell, hey, I belong into the ER, you need to take me there. And so that's what this protein here does. And so it takes it into the membrane of the ER, where the protein translocator is there, or 661. And once it's there, it binds to that 661, and then it continues that translation of the protein. While it's being translated, it's also being translocated inside the ER. Okay? So there are two important things that I would like to take from I would like for you guys to take from this slide. One is that you have the signal sequence here. This signal sequence is what tells the cell that it needs to go into the ER. And then the other one is that the 661, or translocan, it acts as a port that allows this translating protein to go inside the ER lumen, okay, or inside the ER. Once the proteins are able to enter the ER, they get processed. Once they get processed, then they go into the Golgi, and this is what happens. So you have this budding that pretty much forms kind of this capsule that has the proteins that need to go outside of the cell or in the membrane of the cell. And then that then gets shuttled into whatever organelle it needs to go to. And then they get released inside that organelle. And that's how you are able to send all those different proteins to their different destinations. Okay, so now to the work that I do. So I've been working with this small molecule. So we utilize this small molecule to better understand this pathway. And that's what usually in biology you do. You have something that perturbs a specific pathway, and then you ask the question, what happens to the cell? And so that's pretty much what we're doing here. And so this small molecule, it's derived from a natural product. A natural product is a molecule that is being made by a fungus or bacteria, and usually we believe that these natural products are made to defend themselves against other bacteria or other pathogens. But in any case, so we have this small molecule, and through different studies that I'm not going to go into today, we know that it binds to the 661 translocan. So this is the mechanism of action of this small molecule. So this is a normal process that I was just describing to you today about co-translation, translocation. So you have this protein that it's being assembled as is being translocated inside the ER. But when you add CT8, which is this small molecule, that small molecule binds in this pocket here, and that prevents that translated protein to go inside the ER. So it stays in the cytosol, and once it's in the cytosol, there's a machinery called the proteasome that degrades it. So you no longer have this protein. And the inhibition of CT8 is dependent on the signal sequence that I was just telling you about, okay? So that's how this molecule is able to recognize which proteins it's going to inhibit by that signal sequence. So some of the limitations of having this small molecule, at the end, yes, we want to study this process, but also we want to use it as a therapeutic in the future for 661. But one of the problems with this small molecule is it's really, really big, and that it's really, really greasy. And that's a problem when it comes to drugs. Why? Because being that greasy, that means that your cells recognize that grease, and then that it's going to want to break it down. And if it's broken down, then you're really not going to get that compound to go into the target that you're looking for to get an effect on your cells. And so that reduces the drug efficacy. Another thing is that you see all these different stereocenters right here. Those are really difficult to make. So that takes longer for a drug to be made. And so we would like something that doesn't have so many stereocenters, that it's not so greasy. And so for that reason, we decided to do a chemical screen. And so a chemical screen is that you have, in this case, we have 30,000 different compounds, and then we want to find one compound that can inhibit 661 like CT8 does. Okay? So these are the different kind of steps that we went through to try and find a compound that had the same mechanism of action as CT8. And I'll describe them all in a minute. And also, finally, we also wanted to make sure that in fact was inhibiting CT8. I'm sorry, the 661. So the assay development. So we wanna develop an assay that can tell us that a compound is inhibiting 661. And there are different ways that we can go about this. And you need a readout, right? So we knew from previous studies that there's a specific signal sequence, so that it's very, very sensitive to the inhibition of this small compound. And we also know one that is really resistant. So we thought we could use this in our favor so that we can find molecules that were inhibiting 661. Like I said, so we need a reporter, so we need a readout. We need something that tells us usually by light, or there are other different types of reporters, but in this case we utilize light that would tell us whether a compound is functioning or not. And then also we use a chemical library, like I said, of 30,000 compounds. And this is at UCSF. So this is my representation of a protein. And here you have the signal sequence that is really, really, really sensitive to the inhibition of the small molecule of CT8. And also you have this luciferase. Luciferase is an enzyme that catalyzes a chemical reaction that produces light. So whenever you add its substrate, if it's working correctly, then it's going to produce light. So there's going to be, whenever we see something, there's this light, since this is functioning properly and it's not being degraded, then the secretory pathway or the 661 pathway is intact. So the compound is inactive, okay? But if we see no light, then that means that this protein has been degraded and that the compound might be a 661 inhibitor. So then we did what is a high throughput screen. A high throughput screen is just, it's a large assay pretty much, where this is a 384 world plate. And what you can see here, this is on the top. You have very, very, very tiny little wells. The plate is this size. So we have 384 384 compounds that we can add, and we can see whether they work or not. So instead of doing this individually, we just have about 90 of these plates. So that diminishes the throughput. And so we had different controls that would tell us whether the assay was working or not. And so based on this, and this is kind of the process that we did in order to obtain these compounds, to see which compounds were active. This is all done by robotics, which is really nice. You have these robots that dispense the cells, and then they add compound to it. It's really throughput. So this is in summary, this is the results that we got. So every little red dot represents a compound. The blue, those are, this is our positive control. So these are compounds that, well, they're not compounds, but this would be kind of, if red dot is close to the blue line, then it means that it's a hit. It means that it's been inhibiting Sec61. And so the green, that's our negative control. So if the red dot is close to the green, then it means that it's inactive. So out of here, we were able to find 232 compounds that show some inhibition of Sec61, or what we thought was Sec61. And then this translated to a hit rate of 0.7%. Now, there are different other mechanisms that can be translated into an inhibitor of Sec61. One of them could be cell death. And so we wouldn't be that interested in inhibitors if they're just killing the cell. We want something that it's specific to Sec61. And so to kind of go and exclude those compounds that are just killing cells, we have a secondary assay. In that secondary assay, we utilize the very resistant signal sequence to this inhibitor. So in this manner, we can exclude any compounds that are not specific to Sec61. Okay. So to do these secondary assays, we did a dose response. And a dose response is pretty much you have serial dilutions of a compound, and then you see what the response to dose dilutions is. And so then you measure the IC50, and that's the inhibitory concentration at 50%. And so this is just a measurement for us to know how effective those compounds are. That way we can prioritize from compounds that have an IC50 of 1 micromolar to compounds that have that may not be as interested if we have a compound that's, like, for example, 1 nanomolar. So we have this 384 well plate again, and it's just a different way that we set up the experiment, where we just have the different compounds, and in serial dilutions, we did 10 different serial dilutions, and then we test both of these cells to see what the responses were. So here, on the x-axis, you have the IC50 of that dose response, so how effective that compound was against the very sensitive VCAM signal sequence. And then on your y-axis, you have the very resistant signal sequence. And so you pretty much want stuff that it's kind of in this, along this axis, because that means that it is sensitive to VCAM, but that it is insensitive to the very resistant prolactin. And remember, this will tell us any compounds that are not selective towards Sec61. So we started with 232 compounds, and we ended up with just 2 out of 30,000 compounds. So then we just purchased these compounds. We wanted to make sure. Sometimes when you have these compounds and they have been stored in, they're frozen. So when you thaw them, sometimes they can decompose, and so you might be getting a false positive, and so we just purchased these compounds and wanted to make sure that the impact, what we were seeing, was real. And it was. Once again, here we have a dose response of that compound, and we can see that it's only inhibiting the sensitive signal sequence. So it's great. It's looking like it's a Sec61 inhibitor. So then we want to optimize that lead. We want to make a better compound. At this point, our compound was around 3 micromolar. There, it's IC50 against the sensitive signal sequence, and so now we want a compound that perhaps can be better than that. And so to do that, we do structure-activity relationship studies, and this is where the chemists get involved. And so they synthesize and make different variations to the compounds in the hopes that it's going to have a more, that we're going to have a more potent compound. And so unfortunately, I cannot show you any structures today. We're working with a pharmaceutical company, so I'm not allowed to show you structures, but what I can tell you is we have 21 compounds that were purchased based on that initial hit and its structure and different variations of that compound. And 64 compounds that have been synthesized in the lab by 2. One was a rotation student, Kevin, which he was fantastic. And then with another fellow postdoc, Pat. So we've done, out of these SAR studies, we found a compound to have 1 micromolar activity against Sec61. We are in the process of perhaps finding an even more potent compound, but we're still kind of just starting, so hopefully it's going to pan out. And this lead compound is stereochemically more simple. It doesn't have any stereocenters. And then it's smaller in size. It's about 1/3 in size. So size is really important. If you have a really big molecule, then they tend to be really greasy, and that's what we don't want. So having a small molecule as well allows us to can make chemical modifications to it and make it into a better analog without increasing too much of that greasiness. Okay, and so now that we have this lead and that it's somewhat optimized, we want to make sure that in fact it's hitting Sec61 and not another thing. So we have very standardized assays at this point to determine that in the lab. And so first we wanted to check to see what the effects were on the full-length protein. So right now we're just looking at a very small chunk of that protein. And so we wanted to see how it affected the entire protein, because that's the more biological relevant state. And so here you have a Western blot. And so a Western blot is, so these blobs, black blobs that you see here, this is protein. And so we utilize antibodies that are very, very specific to the different proteins that are shown here. For example, this BCAM, that's an antibody that is very specific to BCAM alone. And so this will tell us if a protein is present in cells, pretty much. Tubulin, this is a protein that we just utilize to make sure that we are adding the same amount of protein to each lane, okay? And so in the first lane, that's the DMSO control, so that's our negative control. And then we have our CT8, which is a small molecule that we know already that acts on Sec61. And that's our positive control. And then we have another compound that is very similar to the compound that we found, but that has no activity. And so we just include that as well. And then we just do a dose response once more of 0 dilutions of compound. And in fact, we see that the compound is able to inhibit the expression of the full-length protein. So this is looking good. And so this is, so what we have here, this is some cell lines that a previous postdoc in the lab was able to generate. And these cells have a resistant mutant, so they have a mutation on the Sec61 protein. And so what this mutation does is that it confers resistance to CT8. So once you have this mutation, CT8 no longer can inhibit this protein. And so when we add our compound to the wild type Sec61, so there's no mutation, we see the effect that we were expecting. And then when we add it to the Sec61 that has a mutation, then we see no effect, and that's the red line. So that suggests that the compound is binding in the same pocket as CT8, suggesting a CT8-like mode of action. And then we have the other inactive compound that I was telling you about, and we see no effect. Okay, so for summary, so there are other assays that we've done that I won't talk to you today about, but we're pretty certain that these compounds are inhibiting the Sec61 translocate by some experiments that we've done. And so today I've talked to you about the design and the developed and completed in a high-throughput screen of 30,000 compounds. Out of there, we found one compound that is much smaller and that it's still active against Sec61, and that its complexity is reduced compared to our original compound. And then through a number of experiments, we were able to determine that it's a Sec61 inhibitor. And so for future directions, we want to test this lead compound against other proteins to see if it's selective. We want to see whether we can inhibit other proteins, and if so, which are those proteins. Maybe they can help us understand other diseases that are dependent on these proteins. And then also we are doing another screen of 200,000 compounds looking for inhibitors of another specific signal sequence that is very important for breast cancer. And so with that, I want to acknowledge Jack. He's my PI. He's kind of helped me shape the way I think about science. He's very analytical and has made me be more of a critical thinker. And then I want to thank the lab. This is my boss, Jack, and this is Pat. This is the postdoc that was working on the SAR of those compounds. And then, he's not here. I had a rotation student as well that was working on the derivatization of these compounds. I also want to thank the RACTA Fellowship for funding. This has been a great program for me. I think one of the challenges to go through in science is that sometimes, and at least for me, when I started in grad school, it was like I said, it was a cultural shock. And just having other people around me and having that sense of belonging was really helpful. So the RACTA has definitely brought that for me. And just at the SMDC, this is the core at UCSF that has been the instrumental in these efforts in determining and finding this compound. And so finally, my biggest advice to you, if it was not clear throughout at least the initial part of my talk, is that people have been instrumental in my career or path through science. In one way or another, they have helped me be here, be to where I am. So my biggest advice to you would be to network wherever you're going. Just ask a question. Just, it can be a scientific question. Just make small talk. If you have the opportunity to go to a conference, do so. You're going to learn a lot scientifically, and then you're going to make friends. Like I said, in a conference, that's where I found out about this fellowship opportunity. Network. If you have any other questions, please let me know. If you want to ask me other questions and you don't feel comfortable asking in front of everybody, I'll be here and I'm more more than happy to answer any of those questions.